I was explaining to you about the tumor antigens, which are tumor specific transplantation antigens. Now we will go to the new category of the antigen, which are tumor antigens recognized by xenogenic antibodies. The xenogenic antibodies are nothing but the antibodies that are produced in other animals other than human being. To produce the xenogenic antibodies for the tumors, the tumor homogenate is injected into other animal and the antibodies are produced against the different tumor antigens. One of the speciality of these antigens which are recognized by xenogenic antibodies is that they do not stimulate immune response in the individual where the tumors are developing. Hence, they are called as tumor-associated antigens, in short form TAAs. The tumor-associated antigens are the self-antigens which are expressed by different tissues in the human beings across the race. Since they are common to the human race and they are present in the human being since the development or since his origin, they are tolerized antigens in the sense the immune response does not develop against them because of the tolerance developed against them, because as the immune response develops in an individual after the birth, these antigens being there as common antigens on different tissue surfaces, the immune response recognizes them as self-antigens. Therefore, the antibodies are not going to be produced. Only way the antibodies can be produced is by giving these antigens to other animals as an injection, and then the antibodies can be raised. Therefore, they are classified as antigens recognized by xenogenic antibodies. There are several classes of these antigens expressed on the tissues as well as in the tumors. Generally, in the case of tumors, these antigens are either overexpressed and released into the serum sometimes and can be recognized in the serum of the person suffering from cancer. And these surface antigens can be classified into three different categories. The first major category is oncofetal antigens. The name itself indicates oncofetal, means these antigens are fetal origin expressed in cancer. That means these antigens are present in the fetal stage of the development of an individual and they are essential and perform certain important functions in the fetal condition, and they are once again expressed if the tumor develops in an individual and there is overexpression of these fetal antigens in the tumor. Therefore, they are called as oncofetal antigens. There are two different types of oncofetal antigens. Alpha fetoprotein, in short form it is called as AFP, and carcinoembryonic antigen, which is called as CEA. We will discuss little later in detail about these two antigens. The second category is altered glycoprotein and glycolipid antigens of the membrane. These glycoproteins and glycolipids play a major role in the normal functioning of the cells in a particular given tissue, and sometimes they may be the markers of the tissue origin, and they sometimes get overexpressed as well as few modifications in amino acids may take place in them, but they do not trigger immune response because they are dominant self-antigens. The third category is tissue-specific differentiation antigens. These antigens, as I had mentioned earlier, are expressed during the differentiation stage of the tissue during the embryonic condition, and they may go silent. And once again, if they express due to de-differentiation of the tissue to transform itself into a tumor, their role will be of a different kind here for the progression of the tumor. So we will see now each one of these categories in detail. The alpha fetoprotein antigen is a 70 kilodalton alpha globulin glycoprotein, and it is generally synthesized and secreted in the fetal life by yolk sac and liver, and it plays the role of albumin in the serum of the fetus which is developing in the mother's womb. In the adults, in the human being, the blood viscosity is maintained by albumin, which is secreted by the liver. In the same manner, the blood viscosity in the fetal condition is maintained by alpha fetoprotein. And in the adult stage, in the human being, alpha fetoprotein is present in a very, very low level in the serum. But if hepatocellular carcinomas appear in a human being, there will be significant elevation in alpha fetoprotein levels is found in the serum of the person. It is also observed the AFP levels go high when the person suffers from liver cirrhosis. The liver cirrhosis may be caused due to alcoholic abuse, or it may be caused by the hepatitis B or C virus. And as I mentioned earlier, that hepatitis B and C virus, which causes the liver cirrhosis, may finally lead to hepatocellular carcinoma in the patient. So, AFP is an indicator of hepatocellular carcinoma in a person who was normal up to a particular stage, and suddenly there is an elevation in AFP levels in the serum. So then on diagnosis, it can be found out that the person will be suffering from hepatocellular carcinoma. Similarly, carcinoembryonic antigen, that is the second category of oncofetal antigen, is a 180 kilodalton membrane protein which is highly glycosylated, and it belongs to immunoglobulin superfamily. And had referred to this word immunoglobulin superfamily earlier when I had dealt with the B cell receptors, T cell receptors, and MHC complex. Same way, CEA, carcinoembryonic antigen, also belongs to immunoglobulin superfamily. And it is also observed that it has a major role to play as an intercellular adhesion molecule for the binding of cells. And as you know that when the tumor metastasis takes place, the intercellular binding of the cells in the tumor will be loosened up, and the cells will start migrating from the origin area of the tumor to other areas during the metastasis. So it is observed the CEA levels remarkably increase in the serum of a patient who is having metastatic colon cancer. So it is an indicator of colon carcinoma or colorectal carcinoma. And on treatment of the patient who is suffering from colorectal carcinoma through proper radiation therapy and chemotherapy, as well as probably with the monoclonal antibody therapy, the CA levels go down in the serum of the patient. But when the CA levels increase once again, that will be an indicator that the carcinoma has resurfaced. So therefore, these two antigens, that is AFP and carcinoembryonic antigens, are highly tissue specific as far as indicators of tumorigenesis is concerned. That is, AFP is the indicator of hepatocellular carcinoma and carcinoembryonic antigen is the indicator of colorectal cancer. Let us move on to the next category of the antigen. That is, second category, altered glycoprotein and glycolipid antigens. Sometimes in the tumors, the glycoproteins and glycolipids of the normal cells are abnormally expressed with few substituted amino acids, and these substituted amino acids are not immunodominant epitopes, which I had discussed when I had dealt with antigenic determinants. The strong antigenic determinants or strong antigenic epitopes would consist of glutamic acid, aspartic acid, tyrosine, phenylalanine, and these amino acids play a major role in antigenicity. Whereas in this case, these amino acids are not showing any change. Since non-immunodominant amino acids show some changes, substitutions, and they may show either over-glycosylation, that is increased glycosylation, or under-glycosylation or decreased glycosylation. This is also a change in the property of the glycoproteins or glycolipids. It is not only just change in the amino acids, it could be the glycosylation of these glycolipids and glycoproteins, which also is an indicator of the tumor characteristics. And this abnormal expression of glycolipid and glycoproteins is generally associated with the metastatic behavior and tissue invasion of the tumors. Tumors, when they are highly mature, that is, when they have reached the metastatic stage, they show these altered properties in glycoprotein and glycolipid in order to show movement and migration to other areas which are in fact hostile areas. When a cell of a particular tissue origin leaves its original place and goes to other tissue areas which is a different place and to which they do not belong, it is hostile. In order to accommodate themselves in this hostile environment, in order to have adhesion to some surface area in this hostile environment, the migrating tumor cells, that is metastatic tumor cells, express modified abnormal glycolipids and glycoproteins. But they do not trigger the antibody response in a person because they are not recognized by the immune cells as foreign antigens. One of the example is GD3 glycoside, which is expressed in human melanomas in large quantity. It is a marker glycoside for identification of melanomas using xenogenic antibody. Similarly, it is observed that various human cancers express abnormally high level of blood group antigen A and Lewis Y. See the strangeness here. Blood group antigen is supposed to be expressed on the surface of the RBC. Lewis is another blood group. Similarly, there are Duffy, Kelly, Dexter, Lutheran, MNN blood group. All these are different types of glycolipids and glycoproteins which are expressed on the surface of the RBC and which give the characteristic for the RBC to be detected as a particular blood group. But unfortunately, it is observed that these blood group antigens, instead of not only expressing on the blood group of a person, they are expressed on the surface of some of the human cancers. That is, abnormal expression of these glycoproteins and glycolipids, that is antigen A and Lewis Y, is another indicator that the tissue has undergone transformation. And they may give additional ability for that particular transformed cell for the metastatic process. There is another example, mucin. Mucin is a glycoprotein which is heavily glycosylated and it is expressed in normal cells. It is a self-antigen. Many normal cells in our body express mucin. But it is observed that in the case of tumors, it is underglycosylated or poorly glycosylated. Therefore, it is an abnormal mucin. Mucin expressed with underglycosylation is one of the characteristics of the metastasis, and it is an indicator that the tumor has undergone maturation. Similarly, fibronectin and N-cadherin are other glycoprotein molecules which are generally there in the cells, but instead of N-cadherin, it is E-cadherin in normal cells, and the E-cadherin undergoes modification and becomes N-cadherin, which is the modification of this glycoprotein in the tumors which are showing metastasis. And overexpression of these fibronectin and N-cadherin is observed in metastasizing cells, which facilitates migration of these cells to different tissue areas, other areas of the body. And the molecules that were listed here, that is GD3, mucin, fibronectin, N-cadherin, are used as target molecules for the development of xenogenic antibodies for the identification of the cancer. And they are used for treatment of the particular cancer because they are overexpressed. The monoclonal antibodies are designed to deliver the chemotherapeutic drug to the cell directly by using these antigens. The third category, that is tissue specific differentiation antigens. These antigens are a part of normal cell surface antigens, and they are generally overexpressed during the process of differentiation, of a particular tissue during the embryonic condition, and they are highly specific for a particular tissue, and they are expressed at a particular stage during the normal development of a particular tissue. And these differentiation antigens which are expressed in a particular tissue will be there as markers of that particular tissue. And they may differ from one tissue to other tissue because each tissue differentiates in a different way to perform different function in a human being. So these tissue differentiation antigens once in a while are overexpressed in the tumor, and due to overexpression the tumor undergoes de-differentiation process. I had already explained to you that a tumor cell is a de-differentiated cell and it almost behaves like an embryonic cell. It loses the property of the tissue characteristics. It does not perform the function of a given tissue from which it has originated, just like the way the embryonic tissue, and it has undergone de-differentiation to behave like an embryonic cell, to divide repeatedly, to grow into a large mass of cells, de-differentiated mass of cells. Therefore, the tissue-specific differentiation antigen becomes an important antigen for the identification, and being tissue-specific, the monoclonal antibodies can be produced against these tissue-specific differentiation antigens to target the particular tissue for the treatment purpose. Once again, these antigens do not raise the antibody in an individual. They do not trigger the immune response in an individual when they express in large amount in a given tumor, because they are the self-antigens against which the immune response of the person is tolerized. The malignant lymphomas arising from the transformation of a developing B cell overexpress CD10, CD20 and CD32 receptors. These receptors are expressed by a normal B cell also, and these receptors play a major role in B cell for its function. But it is observed that in the case of B cell lymphoma, in the malignant condition, they are overexpressed on the B cells. And there are now monoclonal antibody therapies against CD10, CD20 and CD32 to target the malignant B cell lymphoma and control its malignancy. It is also observed that in the case of lymphomas, asparagine is converted to aspartic acid, which is first due to glycosylation. The asparagine undergoes glycosylation and then undergoes deglycosylation. Okay, and due to deglycosylation of asparagine, it converts itself into aspartic acid. And this has been observed characteristically in the case of lymphomas. And therefore, it is being targeted. The aspartic acid or asparagine is targeted in lymphomas for the specific treatment with the monoclonal antibodies using xenogenic monoclonal antibodies. It has also been observed that in the case of melanomas, there is overexpression of tyrosinase. And tyrosinase expression is the characteristic of melanomas. And this is as far as the different types of the tumor antigens are concerned and the immune response against them. The first category, that is tumor specific transplantation antigens, are themselves capable of triggering the T cells to develop the immune response against the tumors, whereas the second category fails to do that because they are most of them